everyone, welcome back to my channel. This is lecture 5 of chapter Structure of Chromosomes, Cell Cycle and Cell Division. And in this lecture we are going to basically study phases of mitosis, which is karyokinesis and cytokinesis. Okay, so I am writing, we will study phases of mitosis, which include karyokinesis and cytokinesis. And in karyokinesis, which is division of nucleus, we will study about prophase, metaphase, anaphase and telophase. Okay, and in the end I will teach you a life lesson. Okay, so let's begin. Before proceeding with karyokinesis, let's revise interphase, which we studied in our last lecture. Okay, so this interphase is the preparatory phase for cell division. Okay, and this interphase was divided into 3 substages, which were known as G1, S and G2. G1 is the first growth phase, S is the synthesis phase and G2 is the second growth phase. Okay, now this G1, it was responsible for synthesis of RNA and proteins and also for the increase of volume of cytoplasm. The S phase was responsible for synthesis of DNA and duplication of chromosome. So this was our initial chromosome and it duplicated with 2 chromatids. Okay, and our DNA also got synthesized. Okay, now in G2 phase, more RNA and more proteins were being synthesized. Okay, now let's see how this interphase stage looked under a microscope. So under a microscope, you won't be able to see any such visible change. It's just that the amount of DNA is being getting duplicated. Okay, so no such visible change occurs. This is the cell. I have drawn the animal cell. So this is cell membrane. This is nucleus. In nucleus we have chromatin fibers and nucleolus. This is chromatin fibers. This is nucleolus. This empty space is called cytoplasm. And in animals we also see centrosomes, which are responsible for cell division. So these are centrosomes. And these rays are the aster rays. So they have also got duplicated. Okay, so this whole structure is called centrosome. Centrosome containing 2 centrioles. Okay, so next stage will be our division stage. So this was the interphase stage. Okay, now let's begin with the division stage. And in that we are going to study first about karyokinesis. Okay, let's understand the word karyokinesis. The word karyokinesis means nucleus. And the word kinesis means division. So karyokinesis is division of nucleus. In the interphase stage, our cell already prepared for the cell division. So in karyokinesis, what will happen? This nucleus, this part, it will divide. Okay. First, the division will happen in nucleus, followed by division in the cytoplasm. This cytoplasm will form 2 cells. It will divide from the center and it will form 2 cells. We will study about cytokinesis. Okay, later. Now, this karyokinesis include 4 phases. So, it include 4 phases, which are prophase, metaphase, anaphase, and telophase. You may remember these stages by a very simple mnemonic, which is PM at Kolkata. From Kolkata, remember the word karyokinesis. So, Prime Minister at Kolkata. You may write it in your notebook. Okay. Now, let's first understand the prophase stage. In prophase, we will study about early prophase and late prophase. Early and late. Before that, let's understand what happens in prophase through a very simple mnemonic. Okay. So, you know Popat Lal from Tarak Mehta. So, the mnemonic is Popat Lal can dance superbly. Popat Lal can dance superbly. Write down the mnemonic, okay, below these points in your own language, okay. So, this C stands for centriole move towards opposite poles. Then the next is chromosome, okay. Chromosomes become distinct. Okay, through diagram I will explain these points very simply, don't worry. Now, for D, it stands for this appearance of nuclear membrane and nucleolus. Now, this S stands for spindle fiber formation between two centrioles. Okay, so first what happens, our centrioles means our centrosome containing centrioles, they reach the opposite poles. Then chromosomes becomes distinct, means chromosomes are now clearly visible. In interphase stage they were in the form of chromatin fibers, but now they are fully condensed and they are visible as proper chromosomes. Now, disappearance of nuclear membrane and nucleolus. The nuclear membrane and the nucleolus, they starts disappearing. Next is spindle fiber formation. So the two centrosomes have reached the opposite poles, and between these two poles spindle fibers starts forming. Let's understand this through the diagram. I am first drawing the early prophase stage in case of animal cell. So this is the cell membrane, and inside we have our nucleus. Okay, and this is the nuclear membrane. So inside this nucleus we have our chromosomes, which are now clearly visible. Okay, and we have centrosomes in case of animals, which are now trying to move towards the opposite poles. We have two centrosomes. Now they are trying to move towards the opposite poles. Okay, now let's draw the late prophase, then we will do the labeling. So in late prophase, This is the cell membrane, okay? Inside we have this nucleus containing the nuclear membrane. So we read this point that nuclear membrane starts disappearing. So we'll draw nuclear membrane as disappearing, okay? Now then we also read that these centrosomes, they reach the opposite poles. So now they have reached here. Inside we have fully visible chromosomes. 4 chromosomes are drawn. Okay. Now, next point we read that spindle fibers were being formed between the two centrosomes. So let's draw spindle fibers. Okay. Now let's do the labeling. So these are our centrosome reaching opposite poles. Okay, and these rays are called aster rays. Okay, now these two are the sister chromatids. So these are our chromatids of the chromosomes, and these chromatids are joined at the center with the help of centromere. Okay, now in late prophase you may mark these labelings. Okay, the extra labeling that you have to mark is this: spindle fibers. In the early prophase you are not able to see these spindle fibers, but in late prophase you are able to see these spindle fibers and the disappearance of nuclear membrane. So mark this point also: disappearance of nuclear membrane. Okay, now let's understand again early prophase and late prophase in case of plants. So in case of plants all the things will be same, just that you will not see centrosomes there. Okay, I'm drawing the early prophase stage in case of plant cell. So in plants we have cell wall as the outer boundary. Inside cell wall we have cell membrane. Okay, now here we have a nucleus. Inside nucleus we have chromosomes clearly visible. Okay, this is the early prophase stage. Now you won't see any centrosomes because they are found only in case of animals, and they were responsible for cell division and spindle fiber formation in case of animals only. Now again I am drawing late prophase. Cell wall, cell membrane, nuclear membrane disappearing. Okay, and inside we have our chromosomes clearly visible and spindle fibers. So you may ask this question that in case of animals these spindle fibers were being formed with the help of centrosomes. So who are responsible for forming these spindle fibers in case of plants? The answer is there are microtubules, certain microtubules which are found in case of plants, and that are only responsible for formation of such spindle fibers. Okay, now let's do the labeling. So these two are the chromatids joined at the center with the help of centromere. Okay, this is the nuclear membrane. Okay, this is the cytoplasm. Outer is the cell wall, inner is the cell membrane. Okay, now here two new labelings are this: spindle fiber, spindle fibers, and disappearance of nuclear membrane. So let me write here: disappearance of nuclear membrane. Okay, I hope it is very clear now. Now let's move to the next phase, which is metaphase. Let's understand metaphase. So in metaphase, two important things happen. First is the chromosomes, now they arrange themselves at the center. Okay, they arrange themselves in one plane at the equator. And the second important point is through their centromere, the chromosomes get attached to the spindle fibers. Okay, let's write these 2 points. First point is chromosomes arrange themselves in one plane at the equator, means at the center. Now next point is chromosomes through the centromere, THROUGH, through the centromere get attached with the spindle fibers. Let's understand these 2 points with the diagram. This is the cell membrane, and inside the cell we have our nucleus. But in the prophase stage, we studied that the nuclear membrane has disappeared. So here we will just draw the chromosomes which were present inside the nucleus. No nuclear membrane. So these are our chromosomes. Now, these chromosomes with their centromere, they were attached to the spindle fiber. So let's draw that. First we'll draw this is centrosome. Now we are drawing the spindle fibers. So through centromere, these spindle fibers are attached. This is the centromere. Okay, let's do the labeling. So this is centromere. These are our chromatids. Now this is a spindle fiber. And this is our centrosome. Now let's draw in case of plants. We have cell wall, cell membrane. Now we'll draw the chromosomes arranged in one plane. This is the plane. Okay, equator. Then with the help of spindle fibers, they are attached. Mark by yourself the same labeling in this case also. This is animal cell and this is plant cell. Now let's study the next stage, which is anaphase. In anaphase, what happens? The centromere, it splits and it divides from the center. Okay, and the sister chromatids, after being separated from the centromere, they move towards the opposite poles. Okay, with the contraction of spindle fibers. Okay, now next, what happens in case of animals? A small furrow is formed. Okay, I will explain everything very simply. Now let's write it. So centromere splits. Sister chromatids move towards opposite poles. Then a small furrow forms in animal cells. Why in animal cells? Because this furrow is responsible for the division of cytoplasm. Okay, with the help of furrow, it occurs in animal cell only. In plants, we have cell plate formation. I will explain those things. Okay, so these are the things that occur in anaphase. Now let's understand it through the diagram. Okay, in anaphase also we will study about early anaphase and late anaphase. So let's first draw early and late anaphase in case of animals. Then we will draw in case of plants. So this is the cell. Okay. Now this is the centromere containing one set of chromatid, because it has splitted from the center. Suppose this was our chromosome. From the center it has divided, and so the sister chromatids have separated. And they look like this. We have centrosomes, then spindle fibers. Always join the spindle fibers at the centromere. Okay. This is early anaphase, means the centromere has splitted and these sister chromatids, they are trying to move towards the opposite poles. Now I am drawing the late anaphase stage. This is the cell. These are our sister chromatids who have covered a larger distance now. Okay. Centrosomes containing centrioles. These are the spindle fibers. Okay. One more point which we wrote, that a furrow forms in case of animals. So here we will be drawing a small furrow, means a small depression. Okay, from here this cytoplasm will start dividing. Okay, so this question may be asked that in which stage a furrow formation occurs, okay, in case of cell division. So the answer is anaphase stage. Okay, now so let's do the labelings. So this is the centromere. Okay, these are our spindle fibers. These are our chromatids, which have now separated. Okay, and these are the centrosomes. Now let's draw in case of plants. Now this I am drawing early anaphase stage in case of plants. Cell wall, cell membrane. Centromere has splitted and moving towards the poles. These are the sister chromatids. Okay. We don't have centrosomes in case of plants. Okay. Now, in the late anaphase stage, these sister chromatids have covered a longer distance. So let's draw. Just do all the labeling. This is centromere, this is sister chromatids, okay, and this is spindle fibers. Okay, do the labeling by yourself. Okay, I've explained everything. Now let's study the last phase, which is the telophase stage. In telophase, the chromatids which were moving towards the poles in anaphase, they thin out in the form of chromatin fibers. So let's write this point. Chromatids thin out in the form of chromatin fibers. Okay, now next point, here you will see that nuclear membrane and nucleolus, they reappear. Okay, in the prophase stage they were disappearing, but in telophase they reappear. So nuclear membrane and nucleolus reappear. Then what happens? Spindle fibers, they disappeared. In the prophase stage, they were forming, spindle fibers were being formed, but in telophase stage, they disappear. So, spindle fiber disappear. Now, next point is the furrow which was forming in the anaphase stage, it deepens, okay, and the cell plate is formed in case of plants. Furrow deepens in animals and cell plate forms in case of plant. So, the furrow deepens in animal cell and a cell plate is formed in the center of a plant cell. Okay. This point, first point, second point and third point, these points are exactly opposite of the prophase stage. So this telophase is also opposite of the prophase stage. Okay. Now let's draw the telophase diagram. Telophase, the furrow has deepened. Okay, like this. This is animal cell, and this is the furrow. Okay, and here the chromatids which were moving towards the poles in anaphase, they decondense in the form of chromatin fibers. Okay, and the nuclear membrane also reappears. This is the nuclear membrane. The chromatids have thin out in the form of chromatin fibers. Nucleolus has reappeared. Okay, like this. Okay, and this was our centrosome. Now let's see in case of plants. This is cell wall. This is cell membrane. So here you will see cell plate being formed in the center. This is cell plate, and it will grow towards the periphery. So this is cell plate. Okay, and this is the nucleus. Okay, nuclear membrane, and we have the chromatin fibers, and we have nucleolus. Okay, here you can't see the spindle fibers because the spindle fibers have disappeared. Okay, so let's do the labeling. This is nucleolus. This is nucleus. These are chromatin fibers. Okay, this is nuclear membrane. Okay, cell plate we have marked. This is very important, and this is centrosome. Okay, it is an animal cell and this is a plant cell. Okay, now let's understand cytokinesis. What cyto means? Cytoplasm. And the word kinesis means division. So cytokinesis is division of cytoplasm. It is division of cytoplasm. Our nucleus has already divided. Now it's time for the cytoplasms to divide to form two separate daughter cells. Okay, now so let's understand cytokinesis in animals and in case of plants. Okay, in both cytokinesis occurs by different means. So in animals it occurs with the help of a cleavage furrow formation. So it takes place with cleavage furrow. And in plants it occurs with the help of cell plate. Now let's draw the diagrams. So these are the two cells. Okay, in the telophase these two cells were attached and a furrow was deepening, and that furrow deepened and that separated the two cells. Okay, so inside the cell we have our nucleus and we have chromatin fibers, nucleolus, one centrosome, sorry. Okay, and then here also. Now, see, in case of plants, so we have cell wall, then cell membrane. Okay, in the telophase stage, we saw that a cell plate was being formed in the center. So that cell plate, from the center, it grows outward and joins the cell membrane and the cell wall. Okay, and here the two separate cells are formed like this. Okay, so this is the cell plate which has divided the plant cell into two parts. Okay, and two separate cells are formed. Inside we have the nucleus, chromatin fibers, and nucleolus. Okay, just mark all the labelings. So this was all about cytokinesis. I want to end this lecture by teaching you about surrender. So what is surrender? According to me, surrender is something that we are leaving certain things of our lives which are not under our control, and the things that are constantly giving stress. Okay, so leaving those things in the hands of some higher power and believing that he will definitely take care of it. So no matter what, if you are preparing for any exams, or you are worried about your results, or you are worried about your future, or certain things which are not under your control, just do one simple thing. Just find a place, sit down, just take deep breath 5 to 6 times. Okay, then close your eyes and talk with the higher power which is present inside you. Just have a talk with that power and say that these things are not in my control. I only have control over these things, and I will do my best over the things that I have control. Rest, please take care of it, because these things are really giving me stress. So I am leaving all these things in your hand. Okay, believe me, the moment you will do this, you will instantly feel calmness in your body and in your mind, and you will feel very light. Just give it a try. Then till then, take care of yourself and see you in the next lecture.